Well, good afternoon, everyone. It gives me great pleasure to be here and to speak to you today about genetic research because it's very exciting and a lot of it's happening. I want to thank the organisers for asking me, and I just want to make one comment that program book is the best I've ever seen at a conference yet. It's beautifully set out. I tell you, it's a joy to look at the quality of the paper, and it's beautifully set out and there's nothing missing. Anyway, okay, so now to get back to the genetics. Right, so this is an update of genetics. And there have been two previous Asia-Pacific conferences, and it's delightful to be able to speak at I spoke at those last two, and now at this third one. Different themes each time, but looking forward all the time. Now, I'm going to divide this into two roughly. Firstly, a bit of basic genetics, because I'm not quite sure what everybody knows. And there are a few new little things, not really that much, but then go into the research update. So the basic stuff here is your chromosome, where it all started in 1976. Now, that's not so long ago, 38 years ago, and there's been a huge amount of work in 38 years. But that site on chromosome 15 was localised through cytogenetic rearrangements. And we wondered at the time how much of this DNA, which we couldn't access that easily, was abnormal in here, and a FISH technique picks up something in between. So now, looking at chromosome 15 at that site, it has opened up into an enormously interesting area. And the SNP, the gene, the so-called gene here, has also got an area, I want you to look at this, of SNO RNAs. I'll come back to that. But there are other loci down this end and further loci up each end. So it's a fairly large area. And it has been shown that one end of it was the Prader-Willi end, and the genes on that area have to have a paternal expression. You have to have a father's chromosome functional here, and this is supposed to be pink. It doesn't seem to come out too well. And the yellow ones are not imprinted. So we had the phenomenon of imprinting, which isn't... Prader-Willi was one of the first syndromes where that was described. So that makes it terribly interesting genetically, and there has been a lot of work on that basis alone. The mechanisms leading to Prader-Willi, if you look at a normal person like you and I, who haven't... I won't say we're normal, but we don't have Prader-Willi syndrome. We have a chromosome 15 for mother and father, and the father's one does not work, and the mother's one does. This is, for non-imprinted genes. For other loci, for things like cystic fibrosis, they both work. In Prader-Willi, on chromosome 15, the father's is essential because it's the only one that's working. So if you have a deletion, you haven't got the father. If you have uniparental disomy, you've got two mothers, you haven't got the father. And if you have an imprinting centre defect, you haven't got the father. Now, there are some epigenetic things proposed, and I'm sure they're working, and a lot of people working on epigenetics, which means that the DNA is intact, but this is still not expressing for other reasons, for things feeding into here from other parts of the cell. Okay, the old-fashioned way of doing it was by methylation testing. You had a probe here which didn't cut when it was methylated. You got two bands, and you had in a normal person you got two bands. In Prader-Willi you only got one band, and in Angelman's you got the other. And this was proof positive that you had Prader-Willi syndrome, but it didn't tell you how you got it. It didn't say whether it was a deletion or UPD. This is what the old gels used to look like, and here's a Prader-Willi with one band and not the other, two normals. And here was the chromosomes, in which you, in this case, had a translocation, and here you had a translocation. Now, this translocation led to a deletion of the locus, This translocation was actually UPD. So I don't like seeing, when you hear about mechanisms of Prader-Willi, I don't like seeing it says you can have deletion, UPD, imprinting centre defect, or translocations. That's not the case. The translocations lead to either deletion or UPD. I guess that's being pedantic, but you have to be. Okay, here's the FISH test, which proves you have a deletion if you're missing the probe. Here is a normal 15 with the centrobere and a marker down here in the SNRP region, and here that is missing. So you've done 2 tests, cytogenetics, methylation, we've actually done 3, and FISH. And then you need to do DNA testing to see if it's UPD. And you've got 2 chromosomes the same. The karyotype's normal, but you've got 2 maternal 15s. And if you have a positive methylation test, but it's not deleted on FISH and it's not UPD, then you have an imprinting centre defect. Now this methylation test is so good, 99.5% accurate, there are virtually no other tests that are as good as that. So in that sense, Prader-Willi is way ahead. But by definition, you then have an imprinting centre defect. Now there are some new ways, technology moves all the time, and there are some new ways of looking at that. And you can do methylation-specific MLPA, don't worry too much about what all these things mean. They're just different techniques within the lab. And you can pick a deletion. You can also pick the size. You can pick further probes here as well as the ones in this region. But you can also do methylation. So with the one test, you've got a methylation and you know whether it's deleted and you know the size of the deletion. So some labs are doing this. Other labs are doing microarrays. So I just mentioned that this is a step forward, but it may not be the same for everybody. And the microarrays are also 2 types of microarrays. And this one with the bands across here, it's all actually computer comes into this largely. It'll pick a deletion. And this is the other size. This is the type 2 deletion. The first one, that was the type 1 deletion. You can't really see that much difference here. The other type of microarray is a SNP array, and it will pick a deletion, but it will also pick uniparental disomy, because instead of having a mixture of both mother and father, you've got it segregated, so you haven't got that. So, depending on what you do and how you do it, the diagnosis as such, it has moved forward, but it's very straightforward really, and you can understand it. It does usually involve 2 different tests of some sort, methylation and another mechanism, depending on what the lab uses. Now there's recently a publication of a clinical utility gene card. This is in the European Journal of Human Genetics, and one on Prader-Willi syndrome, and it goes right through all those different ways of doing it, the good and bad points for each one, and hopefully that will lead to a much greater consensus on how to diagnose it, but it's still fairly simple to diagnose. So now we come on to genetic research, which is really the exciting part, and there is a lot of that. Oh, excuse me. You've got work that's going ahead on RNAs, on imprinting, on epigenetics, on mouse models, a new technology, CRISPR technology, the search for targets, and further characterisation of the Prader-Willi gene. Now, I can't talk about all those things in this time. I've just got a few. And Horst Hemke, who is the German specialist in Prader-Willi, he says we haven't actually got a gene as such, a gene that does something. What we have is a gene that controls expression. And there's no point mutations have yet been found in SNRP or on any patient, and they have done patients worldwide. In Australia, we send all our possible imprinting centre defects to Horst Hemke in Germany. SNRP is vital for imprinting, so whatever the importance of imprinting is, that's what SNRP's all about. and the critical region affects the expression. It affects the expression of something. There has to be a target gene. Now, if we look at the locus, the whole area, in more detail, we've got all these genes here. This is the imprinting centre with the SNRP in. You've then got the Angelmans down this end, and in between there are a whole lot of RNAs, and IPW is one that was described quite a long time ago without really knowing what it did exactly. The snow RNAs, a lot of people are researching that, and all these smaller genes down here which are intronless, they all have a slightly different role. None of them accounts for the whole phenotype, and in fact none of these account for the whole phenotype either. Now, if we compare the human to the mouse, it's very similar, but it's not completely the same. And I've always had a problem with mouse models, I have to say. I can't see how the small mouse brain and the small little creature down there can have the same complex abstract thoughts that we have. And I've always had that problem. But anyhow, if you look at the DNA, the mouse is still very useful because you can experiment with it, but there are some things that we have that they don't have, and there are some things that they have which we don't have. Nevertheless, a lot of work is done on mouse, and I just want to go into it a bit. In fact, SNRP was discovered in the mouse, and it's on their chromosome 7, which is syntenic to our 15, and it's also got paternal expression. And we thought that it was very valid to use mouse for the study of the genetics of Prader-Willi because there's so many similarities. However, when you started to look at what the mouse models were, it wasn't quite that simple. If you just delete in the mouse the whole locus, strangely enough, some of them died after birth. What have I done? How do I get rid of that? How do I get rid of that? If you get rid of the whole locus, some of the mice died after birth before weaning, but then the ones that didn't die were not obese and they were actually fertile, so they didn't really have Prader-Willi syndrome. And if you started to knock out each of those genes that I showed you individually, you got different results. The SNRP one, if you knocked out, was a normal mouse. The SNRF knockout was a normal mouse. The Mackinac N3 was a normal mouse, but the snow RNA, one of the ones that's very commonly used, is an abnormal mouse. The Nectin knockout is an abnormal mouse. So the results were inconsistent. And if you look at the mouse, each of them had a different feature that was wrong with them. In the Garvan Institute, currently in Sydney, they're working on the SNORD 116 mouse, and SNORD 116 was in the centre, roughly in the centre of that region. I'll show you that again. But these mice are all different. So the mouse model has its limitations, and a review of the mouse models in Prader-Willi syndrome came out from the Garvan Institute. This is an excellent review. And it just took each of the models and said how was it generated, what strain it was, what it could do, what you could do with it. And it came to the conclusion that in this review they described which mouse models have been generated so far and the similarities to the human features. But the conclusion is that there's no mouse model which is actually that good for Prader-Willi. And I want to say that yesterday at the foundation, not yesterday, the day before at the foundation meeting, Rob Nichols is developing a pig model for Prader-Willi, which he says is going to be so much better. And it hasn't yet been thoroughly worked out. He's getting a farm where he can harvest, and he can grow pigs and harvest them and do experiments on them. And he said it's a much better model for Prader-Willi. So we've got a huge future ahead of us in pig chromosome studies on Prader-Willi. So now if we just look at the RNA, the DNA, this is the classic algorithm. The DNA produces RNA, the RNA is modified from the DNA, and in that cell it goes to protein. Now this whole area of RNomics, which I think is fantastic, There are now so many types of RNA that it's getting to be quite interesting, and I actually, for the first time, just recently heard of TASI RNA. But I'll quickly work you through. There's messenger RNA, transfer RNA, ribosomal, pre-ribosomal, pre-transcription, so various stages here, and they all express what is going to come out at the end. You've got SNO RNAs, and that's what that's the ones that I showed you, and the SNORD 116 is a SNO RNA. MicroRNAs, the long non-coding RNA, and that's IPW, is a long non-coding RNA. And you've got double-strand small interfering RNAs, which get in and interfere with the action of the others, and transacting small interfering. And the easy ones are the epigenetically activated small interfering RNAs and short guides. So it's amazing. Every time you pick up a genetic journal, there's another type of RNA, and I think that's fantastic and really interesting. But I can't tell you much at this stage about what they have found with each one. Now, looking for targets, this is something else that I think is a real breakthrough. A paper came out last year in Nature Genetics talking about, the title was Crosstalk Between Imprinted Loci in the Prader-Willi Syndrome. And apart from the initial blurb, it said that in this paper it demonstrated that these people looked for the whole genome to see what maternally expressed genes there were, and if they could find some new ones. And they did find a new one, and it was in the non-coding IPW RNA. It identifies a critical role for that in modulating the expression of a maternally expressed gene in trans on another chromosome, in other words, and I'll show you that here. And I've called it Have We Got a Target? To be a bit provocative, to show you it's a real breakthrough. I'm not saying that this is a target, but it's showing you that it's possible. Here's chromosome 15, here's our RNAs, here's IPW, and in this paper, because this is paternally expressed, they show that IPW goes into the cytoplasm, picks up another molecule and affects the expression of a gene on chromosome 14, something quite different. So that has got to be explored. So that's really a good breakthrough, I think. Now, another new technology which is a breakthrough is CRISPR, and that stands for Clustered Regularly Interspersed Short Palindromic Repeats. Okay, and these are very important, and they are new. It's only new, but you can create mutations in cells very specifically, not just any old mutation like of a big area where you might delete a big area, which has been known already, so that's not really new, but these are extremely specific, and you can do a lot with CRISPR cells, and I think it just shows you that within 2 months of first hearing about CRISPRs, the next thing you get is an email to tell you that if you buy it from this place, you can get into a loyalty program. Well, that means, that has got to mean they're here to stay. Anyway, so here, I want to summarise here to say that I'd love to spend much more time talking about each of those, but there isn't time. But there's certainly a lot going on in genetic research, and it's all very exciting. Thank you.